The biology role model I chose to research was Elizabeth Blackburn. She was born in Hobart, Tasmania in 1948. From a very young age, she enjoyed exploring nature and became very curious about the existence of living things. Both of her parents were family physicians who encouraged her curiosity and pushed her to continue to explore her many questions about the nature of life. Professor Blackburn earned her Bachelor of Science and Masters of Science degrees from the University of Melbourne and her PhD from the University of Cambridge in England. She later completed her postdoctoral work in molecular and cellular biology at Yale University, and she currently runs her own lab at the University of California, San Francisco. Blackburn's main field of study is molecular biology. In particular, she focuses on telomeres and telomerase. In her early career, she studied tetrahymena, which is pictured in the top right, which is a single-celled organism also known as pond scum, because it had many small linear chromosomes with an abundant amount of telomeres. She noticed the telomeres of tetrahymena did not become progressively shorter after each successive cell division. This led her to the discovery of the enzyme telomerase, which is responsible for the maintenance of telomere length. In 2009, she won a Nobel Prize for this co-discovery. Throughout her journey to discover telomerase, she faced many dead ends and had to overcome many obstacles. She highlights the importance of persistence in research, as she said, "Experiments don't work much of the time. The very easy ones have been done, so when you do things, you know that there will be a certain number of times things just won't work. You keep trying, you try a different way." I admire Elizabeth Blackburn's drive and dedication for delving into telomere research and not giving up till she discovered telomerase. Additionally, as an inspiring researcher, I admire and appreciate her encouragement for future generations to seek answers to the many unanswered scientific questions. I identify with how her interest in science was sparked at a young age by her parents' involvement in the medical field. For me, growing up near Mayo Clinic and my mom being a doctor sparked my interest in science and research. And as a young woman, I admire her success as a researcher and hope to pursue a PhD to conduct my own research one day. Professor Blackburn's two main hobbies include playing the piano and Sudoku. She obviously thoroughly enjoys her research career, as she once said, "People who love their work don't need hobbies. Work is your hobby." In her most recent paper, Elizabeth Blackburn investigated the relationship between adverse childhood experiences and leukocyte telomere length in individuals with major depressive disorder. The study followed 20 unmedicated individuals with major depressive disorder and 20 healthy individuals. The healthy controls were each matched to an individual with depression based on age, sex, and ethnicity. The adverse childhood experiences of both groups were documented through retrospective self-reports, and blood was drawn to measure the leukocyte telomere length and resting telomerase activity. In healthy individuals, the researchers found a correlation between adverse childhood experiences and shortened telomere length in adults. However, for the individuals with depressive disorder and greater adverse childhood experiences demonstrated increased telomerase activity but showed no correlation to telomere shortening. Therefore, the activation of telomeres in individuals with depression and adverse childhood experiences may possibly possess a compensatory response to maintain the length of endangered telomeres. A defining moment of Blackburn's research career was her discovery of the crucial enzyme telomerase for maintaining telomere length. Throughout her research career, Professor Blackburn has continued to further investigate the association of telomere length and health and aging in several capacities. She and her team found that the more chronic stress an individual is under, the shorter their telomeres. She also noticed the social aspect of telomeres as life experiences and how one reacts to them impact telomere length. Things such as emotional neglect or exposure to violent experiences early on in childhood impact telomere length later on in life. She emphasizes that telomere science sheds light on the interconnectedness of humanity, as we all have the potential to impact our own and others' telomeres. In her lab at the University of California, San Francisco, she continues to investigate the extent to which humans can potentially impact their telomeres and shape both health and disease span throughout one's lifetime.